Hello, I'm Frederick Maisha, a Swiss physician. I've been very intrigued by the composition of the cell nucleus. I've been collecting pus from bandages of my patients, and I seem to have found something. Through experimentation, I was able to isolate this unknown substance in the nucleus. This substance happens to be acidic, and it also contains a large amount of phosphorus. I think I'll call it nuclein. Hello, I'm Frederick Griffith. I'm a medical officer for the British military. I've been tasked with the research of a pneumonia disease that is plaguing Europe. Using 2 different strains of pneumonia bacteria, I've been injecting mice with them to see how they react. Let's first try the S strain. As you can see, the mice died here. Let's move on to the R strain test. It seems that the mice are still alive. I wonder what would happen if we add a dead S strain. Seems that the mice is alive. What if we added a dead S strain and an R strain? What would happen to the mice? Seems that it killed the mice. Somehow, the hereditary substance from the dead S strain killed the first mice had passed down to the R strain. I believe I will call this transformation. Hello, my name is Georgine Hammerling. I'm a biologist and I'm researching where the genetic info is stored in the cell. I've been conducting experiments using green algae. I've removed the nucleus from the algae, and as you can see here, it has stopped regenerating. I believe that the nucleus controls the development and genetic material of the cell. Hello, I'm Oswald Avery, a physician and medical researcher. I'm here with my coworkers Colin McLeod and Macklin McCarty, and we are trying to confirm if DNA is the transforming substance for cells. We are using Streptococcus bacteria, which has S strain and R strains for our experiment. I've been treating a dead S strain with different enzymes that destroy different substances, and I believe I made a breakthrough. When adding this dead S strain without protein, you can see here when mixed with the R strain bacteria, it transforms, which means that the protein did not carry over the genes. When adding this enzyme that destroys DNA, you see that the R strain no longer transforms. This is confirmation that DNA transformed the substance and not the protein, although I must conduct further experiments as I am not sure if all the proteins have been destroyed by the enzymes. Hello, I'm Erwin Chargaff, an organic chemist, and I do not agree with other scientists that the proportions of nitrogenous DNA bases stay the same for every species. I was able to discover that there are actually fixed ratios for bases. For example, adenine and thymine always have the same equal amount, while on the other hand, guanine and cytosine have the same amount. Take human DNA for an example. Guanine and cytosine have about the same amount of about 20%, while thymine and adenine have the same amount of about 30%. This shows that adenine and thymine have a ratio of 1:1, while guanine and cytosine have a ratio of 1:1. I think I'll name this discovery after myself. Chargaff's rules. Hello, I'm Alfred D. Hershey. I'm a bacteriologist. I'm here with Martha Chase, and we are doing experiments to determine if proteins or DNA function as genetic material. Here we are using E. coli bacteria and a virus that infects bacteria, also known as a bacteriophage. This virus contains both DNA and a protein code, so we are going to infect the bacteria to see if the DNA or protein code causes the transformation. We are using radioisotopes of phosphorus and sulfur, shown as P and S. Sulfur is labeled protein and phosphorus is labeled DNA. Depending on which shows radioactivity, it should mean that it entered the cell. Our results indicate that the bacteria infected by the virus containing DNA shows radioactivity. This is conclusive data that DNA must be responsible for carrying genetic information.
Hello, I'm Rosalind Franklin, an English chemist and X-ray crystallographer. I've been using X-ray crystallography to study the shape of the cell. I noticed that the image produced by the X-ray was in the shape of an X. Analyzing the pattern of the crystallograph, I propose that the sugar phosphate backbones of DNA are actually facing outward rather than inward. I also propose that the DNA is structured in a double helix that rotates clockwise. I was able to identify that the diameter of the DNA molecule was 2 nanometers, and the length of one turn in the helix is 3.4 nanometers. Although I'm not quite sure how the nitrogenous bases are connected in the middle of the helix.
Hello, my name is James Watson, an American biologist and geneticist. I'm here with my partner, Francis Crick, and we are trying to build a model of DNA molecules, with Maurice Wilkins revealing details about Franklin's findings, such as the double helix. I believe we can construct a model that incorporates her information as well as everything else we know about DNA. In this model, we see that the phosphate backbone is represented in yellow, guanine in green, cytosine in pink, thymine in blue, and adenine in purple. It also shows that the nitrogenous bases are connected by hydrogen bonds. Thymine is bonded with adenine with 2 hydrogen bonds represented in white, and guanine is bonded with cytosine by 3 hydrogen bonds represented in gray. Hello, I'm Kerry Mullis, an American biochemist, and I created the polymerase chain reaction, or PCR. This technique copies millions of DNA which amplifies it so it can be analyzed. We first heat the sample, denaturing the DNA, then synthesize it with an enzyme. This process results in the DNA duplicating, leading to millions of exact copies. Without PCR, it would be difficult to study isolated segments of DNA. Hello, my name is Ian Wilmot, an English biologist. I'm the leader of a group of scientists and researchers who cloned the first mammal of an adult cell. The cloned sheep named Dolly was created using somatic cell nuclear transfer. She was bred from 3 products provided from 3 other sheep. One provided an egg, the other DNA, and another carried the embryo. Using an egg and nucleus from 2 sheep, we used electricity to fuse them together and to stimulate cell division. The new divided cell was placed in the 3rd sheep to develop, and Dolly was born. Dolly is genetically identical to her mother, who provided the nucleus. Why was Rosalind Franklin not part of the group that was awarded a Nobel Prize in 1962? While she had died of cancer 4 years earlier, she would not have been able to go receive the award. The Nobel Awards given to those already dead were rarely given at the time.